Caterina Villari, Associate Professor of University of Georgia. Welcome. Thanks for joining today.
Thank you. Thank you for having me, Demian.
Absolutely. I'm really excited to talk about this today. We're going to be diving into molecular tools for diagnosis and surveillance of plant pathogens, specifically talking about one particular method that your lab has been working a lot, which is called LAMP. And it's kind of like a little bit different from the typical, PCR that we usually would hear about. So before we dive into that, please start by talking about your lab. What are your main research projects or research lines that you guys work with?
Okay, yeah. So in my lab, we work in different direction. Different members of the lab work on different projects, but I work a lot with host-resistant mechanisms. So the metabolomic part, how do trees respond when there is a pest or a pathogen, all their defense mechanisms, including secondary metabolites, mostly secondary metabolites. And also a little bit of resistance selection. How can we use this chemical response to help us identify resistant families to a certain disease or to a certain insect group? And this is one component of the work in my lab. The other, it's a lot on diagnostics. And so we develop tools to advance diagnostics and possibly bring it out of the lab, like portable tools like the LAMP that I'm going to be talking today, and other more sequencing-based tools like a little bit metabarcoding, or right now we're trying a little bit of nanopore sequencing. And so looking more at the whole community of microbes associated with a type of symptomatic or non-symptomatic tree. So completely different direction, more defense response based and chemistry based, or more molecular. So we kind of tackle a little bit of both those aspects in the lab.
So take us a little bit to the beginning, and this is going to be a very philosophical question, but how do you end up working in forest pathology?
That's a big one. I always knew I liked the natural resources, or STEM. I was actually unsure if to go in physics or forestry, so those were my two choices when I started college, and I ended up forestry. I think I would have liked physics as well. I tend to get passionate on whatever I do. And with the forestry, when I started the forestry school back in Italy, I graduated. I did all my education and graduated in Italy. I quickly realized I love the forestry world, but I really love the forest biology part. And even though I did it and enjoyed it, all the forest business, all the procurement part, it was interesting, but it was not my gig, let's put it that way. And so with the forest biology component, I liked forest health a lot, both entomology and pathology. I was in contact with both professors at the time, and I needed a senior thesis project, and I kind of inquired where there was availability. I could have ended up in a forest entomology lab. There was a space in forest pathology, and that's how I started. I did my senior thesis in forest pathology, and then I kept working with the same professor as a lab worker for a while, and then I did my master's with them, and that was my beginning as a pathologist. But again, forest health in general has always been my passion. So even my PhD, it's actually from an entomology department in University of Padua, working on fungi associated with insects. So I always worked at the interaction between insect pathogens and the host plant. So it's kind of, they're different departments, yes, but it's all one thing, forest health.
So let's start talking about diagnostics, right? So when we think about diagnostics in forest pathology, for example, there are different methods, even before we get into the details of LAMP, but can you talk a little bit about the different options that we have when we think about diagnostics in pathology?
Yeah, sure. So, well, the first thing you could do diagnostic with is just with your eyes. I mean, you are in the field, you recognize symptoms or signs of something, and sometimes if you're lucky, there are certain diseases that you're really able to just diagnose just by looking at. They're certain it's like, well, this is it. There's no need to do further analysis. But most of the time, that's not the case. And many symptoms are overlapping, are similar. Many diseases will cause the same symptoms. And so there's very often the need to do a little bit more of investigation. And so what I would do in that case is collect samples. And from there, you can go either with the molecular testing way or the classic isolation method. So you bring the tissue back and you try to isolate the fungus out of it to have a better idea of what's going on. Isolation are still a very powerful tool. I don't think we could go completely without it, but they are very time consuming, and not all pathogens likes to grow on the same type of media. So sometimes there are many fastidious organisms that will never grow on media to start with. And so just relying on isolation methods is just not an option anymore. It's still a valuable steps and process that it's good to keep using, but you always, most of the time, have to do it beside molecular, together with molecular methods. And so if you're lucky enough, you could do molecular method directly from plant tissues, but many times you will have to isolate and then do molecular methods on whatever isolates you have. But all of this is very time consuming. So that's why I like to try to develop tools that could kind of shorten this timing and bring the diagnosis out of the needs of having a lab associated.
Yeah, and that's where kind of like LAMP gets into the game, right? So can you speak about like what exactly LAMP is?
Yeah, so LAMP stands for Loop-mediated isothermal amplification, which sounds like a complex name, but at the end of the day, is just amplification of DNA. So nothing very dissimilar from classic PCR, where you have amplification of DNA at the end of it. It's just that it works with a different type of chemistry. And so because of the characteristic of the chemical reaction that happens and then the enzymatic reaction, it's more suitable to be brought directly in the field because you don't require cycling. Now, for normal PCR, for PCR to work, you have to have the cycling. So a change in temperature, cold to hot, basically. And that requires specific machinery that could do that. While LAMP works at the same temperature, the enzyme can work straight away, doesn't need to have like the opening of the separation of the two chains of the DNA. So it can work as it is. And so you just need something that keeps your sample at 65 degrees, which is very achievable. It's just a hot plate at the end of the day. And the enzyme works by itself. And at the end, you have a lot of amplified DNA. So that's what your end product of LAMP is. Amplification of DNA, no different from PCR, other type of PCR, but it just worked with a different enzyme. You have a different type of primers, but your final product is the same. A lot of target amplified product of your DNA of your target, whatever organism you're searching for.
Gotcha. And so basically this is still not one type of PCR. This is a completely different method, just like runs similar to what a PCR is, right?
Yes, it's not, we cannot call it PCR. It works with a completely different enzymes, and the reaction, if you like look at the little video, what happens when you have the LAMP reaction, it's completely different from the classic PCR amplification. But the final product, it's something equivalent. It's a lot of amplified DNA of whatever you're searching. It will have different shapes. It doesn't come in single like little band. It has the final product. It's a mix of different dimensions. It's called cauliflower structure. It's kind of three-dimensionally very complicated. But from a user point of view, all this doesn't really matter because you don't have to understand exactly what shape it has. You just have a lot of your target DNA, and if you use visualization method, you're just going to see it either as a fluorescent light, either at a color change in your reaction, and you don't care what chemistry is happening. What you care is that you're able to detect that huge amount of product that has been amplified and without the need of a lab, basically.
So yeah, and that's basically one of the big benefits, right? You don't need a specific lab. You don't need that thermocycler or a specific machinery to run the process. What are other benefits about these methods besides, not really having to do that on the lab? Is it more accurate, for example, or?
Yes, it is. It can be very specific because, and here I'm going to get a little bit more technical, but normal PCR or real-time PCR starts with two primers, forward and a reverse. And so you have two regions of about 20 pair of bays per primer where you can search to maximize your specificity. So searching for regions that are conserved in your target DNA, but they're different from whatever you don't want to amplify. And so you have that region there, and even of the primer, not all the length of it is very useful. So you have a very short room to have specificity, to exploit your specificity. While with LAMP, you use up to 8 different regions to design your primer set. And so you have a lot more opportunity to take advantage of any potential, say, mismatch that it's between your target region and something that you don't want to amplify. And with that, you can really enhance specificity. You can achieve way better level of specificity than other primers, just because you have more room to search for those mismatches, and up to eight. So that's 4 times more.
More specific. So even when you have this sort of specificity using LAMP, is there still room for non-target amplification, or is that something that typically we wouldn't see in something like LAMP?
No, you always have the risk of non-target amplification. That it's all enzymatic reaction. None is perfect for how accurate you try to design it, but it's usually pretty strong. So that it really depends on how you have designed your primers. It's not, you have to be careful of how you select your mismatches. Not all mismatches are as strong as others, so you need to be careful of, and that it all goes at the design stage. So those are things that my lab would do, but an end user would not need to deal with. But all the
go ahead. Sorry.
Yeah, so it's on the design phase, you really have to pay attention on how you position your primers. And then another thing that sometimes we don't realize, that if we design primers on some of the common regions like ITS, we know the one that are sequenced and deposited, but there might be other copies out there in the genome that we're not aware of, and then it can cross-react. So it's always good to validate. I mean, you always have to validate your assays with a big array of non-target to make sure it doesn't really amplify anything else.
Yeah, exactly. I was going to ask about that. So when we think about the process of, developing this LAMP method for any particular disease, you always start, of course, developing those primers. But then between that and the validation phase, how that process actually looks like?
Yeah, so in a routine like development of LAMP assay, you start from the computer. So you get the sequence of your target, you try to align it with as much similar organism that you can. You really build like a phylogeny tree with whatever is in the surrounding, and you try to kind of make sure you have representative of all the closest species there, and you start on the computer in silico. Then once you have your primer, you really have to test it in vitro. There's no escape from that. So making sure to validate the primer set that you have designed in a as big as collection as possible of organisms, and it's not just random, like I work with fungi mainly, it's not just random fungi. It have to be those that are taxonomically more closely related with the fungi that you want to target, and you want to make sure it doesn't cross-react with the close siblings, let's call it that way. And other very important thing to test is also any potential other organisms that it's possibly present in your host plant. So if you're designing something that it's for pine needles, then you want to make sure you test any other pathogen or any other organism that might be potentially in your pine species needles, because you never know. You'll have to test. And another very
you're going to run into those for sure. You're going to run into those. Yeah.
And sometimes people forget, test your host plant, because it might cross-react with your own host plant, and that's, of course, problematic. And so all those validation steps are very, very critical. But that's for any molecular test, even normal PCR. This is common practice for all of that. It doesn't change for LAMP. You do exactly the same validation step that you would do for any other molecular primers-based technique.
And what kind of pathogens has this technique been used or being researched in the last decade? I know that you were involved in things like laurel wilt and more recently pitch canker. How has that experience kind of like been with LAMP?
Yeah, so LAMP has become more and more popular recently. When I started working with LAMP, very, very few were doing it. I learned in UK at the time, and it was kind of very innovative. Very few people would work with it. But in the forestry world and forest health, it has slowly gained traction and attention. So I'm not, of course, the only lab working with LAMP. There are many others, and especially in Europe, they're very, very active with it. That's where I started as well. That's where I learned it first time. So UK has been developing several probes. Sweden, Italy. There's many, many different countries that have worked on LAMP primers for forest pathogens or forest system. And so we have, I know they're out there for ash dieback, Heterobasidion, there is for Xylella. We developed the one for pitch canker. We developed one for brown spot needle blight and the 2 Dotistroma species. There are some for insects, forest insects. There is for pinewood nematode for sure. And I don't remember all on top of my head, but there is a very long list. So it is something that has gained traction in its in forest pathology, and more it's getting used. So and more there will going to be need for it, because I mean, people saw the use of it, how it's conveniently brought to the field, or even when you use it in the lab. I mean, most of the direction we do it in the lab, we don't necessarily bring the little tool out in the field, but it's for us it's convenient because it's very rapid. It gives you a result in about 20 minutes, which is really a big improvement if you compare with real-time PCR that takes up to 2 hours and a half. And the sensitivity of LAMP is comparable of that one in real time, so it's more sensitive than normal PCR in a quarter of the time. So that's a big advantage for us, so that we can like run a lot of samples in a little time. And one thing that I forgot to mention, because it's meant to be field portable, I mean, one of the very good characteristics is that it's very resistant to inhibitors. And so you can use crude DNA extract. You don't have to have clean DNA, which sometimes is what takes a lot of time to get, like extracting and purifying your extract. It's time consuming. While LAMP, we use a protocol that was developed by an Australian group, and it's 30 seconds of extraction. It's like so quick, and it's raw extract, it's fine, but for LAMP it works. And so in 30 seconds you extract your DNA, and in 20 minutes you get the reaction. So that's very rapid, and so that's to us a great advantage.
So basically 100% you can do this in the field.
Yeah, it's meant to be. The extraction is pipette free, the reaction can be pipette free. It's all based on paper dipsticks. And so you go in the field, there's several brands that produce those, so you don't have to stick to the brand that we have, but we have a little instrument like this. It's called Genie. It's like literally, this is the dimension, and we can bring that in the field with us. It's battery operated. All the extractions is pipette free. You bring your reaction strips, you get the sample that you want to test, you extract in 30 seconds, a minute is really, really rapid, and you run it there in the field, and you have a response, yes or no, in 20 minutes. Now, it only will tell you yes or no. You cannot do any further processing out of that. So that's one of the limitations of LAMP. You cannot sequence, you cannot do any post-production work. Just going to tell you yes or no. It's your target organism here, yes or no.
Well, yeah, but that still has massive applications when we're talking about diagnostics, when we're talking about invasive species, when we're talking about, early detection or rapid identification in different contexts, right? Not only in research, but surveillance, professional arborists, landowners. It has a big range of applications for sure.
Yeah, no, definitely. And just being able to confirm the presence or the absence, like of an invasive species or a quarantine species, that's very, very important. This technology, I mean, it's not, of course, forest health has been one of the application, but it's very much used in veterinary science, in medicines.
Yeah, I was going to ask if like, where was this technology actually developed? I imagine this was brought to forest health, but also maybe in crop science as well. But in what field this was actually developed?
The first time I heard about it, it was in veterinary, but I think it was developed like in biology, human health, and veterinary. Yes, that's
interesting.
That's the first paper that I remember out of it.
Yeah. And what are some of the projects that you are kind of like start looking into or actively working regarding to introducing this method in terms of diagnostics?
Yeah, so right now we are working on oak wilt. We're trying to develop a oak wilt probe, and we're almost wrapping up with that. So hopefully it will be available soon. And so this is one of the latest one that I'm working with one of my students, actually two of my students. But I also, at the same time, I'm also working in a non-forest related field. One of my students is from wildlife sciences, and she's developing a probe for the detection of neurotoxic cyanobacteria that grows on an invasive weed. And so completely different application, but still very useful and powerful. So we're trying to see if we can detect the cyanobacteria before it becomes, before it start producing the neurotoxin. And this is at the base of a very important disease that it's Up to the chains, it goes up to eagles. So it's a very
Interesting.
it's a problematic issue here in the
Yeah.
South. Yes.
And, thinking about the role of LAMP as, how it's, or how we traditionally approach forest health issues, forest health diagnostics, I'm wondering, how do you see this tool kind of like being implemented in, future decades, right? Right now, it's something that is being recently started to be developed, implemented in some pathogens, in some areas, some insects that you were mentioning. But how do you see this kind of like moving forward in the next couple of years?
Yeah, so the thing of LAMP, and when I try to talk of LAMP with newer colleagues, or when I try to even with the funding agencies, is that let's say the work that goes into creating an assay, it's a lot. It requires a lot of work, a lot of testing. But once the assay is created, the actual implementation of it, it's very easy. It's very user-friendly. And so you don't need particular training to be able to perform LAMP. And that's what I've been trying to do. So ideally, that would be my hope. We could train professionals in being able to do the reactions. As anybody have learned to do a nasal swab when we were in COVID time, it didn't take a PhD to be able to do one of those reactions. LAMP is very similar. Once you have all, of course, it takes a lot of work to get to that stage, but once you're at that stage and it's all developed, it's very, very easy. So I'm actually performing a lot of trainings on the techniques to professionals. We've done a couple of workshops. They were funded by NIFA, and we're trying as much as we can to show to practitioner how easy it is, so that if they learn, then they could be the one running it without having to ever get back to me. Just buying some of those kits are already commercially available, and we're in contact with some of those companies. So the idea would be, say for the oak wilt, once we have developed it and published and it's validated and everything, we could work with some of those companies so that they would produce the kit, and those kits could go directly to arborists, to different city municipalities, so that they never have to get back to me. They could do it themselves. I'm happy to provide like a one-time training once in a while, and then people go and do the test by themselves. And so this would improve so much surveillance in cities if we're talking of urban diseases like oak wilts. But think of if people learn to use it at port of entries, where you could screen for invasive pathogens, or even invasive weeds, seeds and stuff, anything that you don't want to get through, and then you can detect in a molecular way. It could be screened by port of entry inspection officers.
Is there any example I can think of particular area? It doesn't have to be specifically for forest health, but that this technology has actually been implemented into commercial kit that people or a specific institution will use?
The Phytophthora ramorum test, I think, in UK has been commercially available. There are companies that you could buy, like the kit for the Phytophthora ramorum. It was a big problem in UK. That's what they were working on when I was there learning the technology. They were working a lot on this Phytophthora ramorum detection. And I think to remember that there were already some lateral flow device with the LAMP primers all loaded up that you could buy and you could do yourself in the field, basically, all prepared. And so I'm pretty sure of that. I don't remember on top of my head of others in forest health world. Of course, if you go at other, like COVID, those are already available, and you can buy them. But in forest health, I pretty much remember the Phytophthora ramorum one, so sudden oak death, causal agent, and I don't remember of others, but they're right now trying to, for instance, incorporate our assay on the Lecanosticta cicola, so the causal agent of brown spot needle blight, as one of the procedures recommended by EPPO in Europe for testing of the pathogen. So if it becomes like recognized even from agencies like EPPO, possibly people will start to implement it more. And I was aware of that because they reached out to us to kind of ask some clarification to make sure it made sense. But I'm sure if they reach out to me, there are probably other assays for other pathogens that are probably being included in the EPPO recommendations for diagnosis. And so that would be a good actual implementation of it.
Yeah, and even thinking, this is kind of like a one step further thinking about the impact of this technology around the world, but thinking about countries where traditional costs for diagnostics with PCR or even qPCR are kind of like prohibited, right? Places where it's not always accessible to do these kind of like identifications. This could help a lot for early detection or surveillance or things like that.
Yeah, definitely, because like there are different ways of doing LAMP, different ways of visualizing it. The one we use this portable tool, but you could run LAMP without that if you want to. All you need at the end is a hot plate, and so there are many developing countries that are using it with the colorimetric assay version, in which you really only need hot plate. So that is way cheaper, first of all, and it's you don't really need much technology there. And then all you have to do is look at, like with your naked eye, you see a change in color, usually from yellow to red or vice versa,
Exactly.
and you can assess the assay that way. So yeah, it's very much used in many countries. When and this was back at my PhD times, so a little bit ago, but a colleague of mine who was in the veterinary world, and he was using LAMP a lot. He was doing training in Africa at the time for the use of it, because it was a technology that they could use there in their veterinary clinic to quickly diagnose, I think was some epizoic problem. Not veterinarian, don't remember, it's been ages ago, but I remember that he was saying, I'm training them for LAMP because with like barely minimum equipped lab, this is something they could do, because especially with the hot plate version of it, all you need is a socket of energy. And sometimes you could have battery period at once. And so that's all you need. And so this is really helpful for those places where you might not be able to have access to a real-time PCR, fancy machinery and stuff like that.
Yeah, absolutely. Well, Caterina, I want to thank you for joining and explaining all of these. I think this is very interesting technology, and hopefully we're going to see that being implemented more and more over the next couple of years. Of course, we're only scratching the surface here on how this technology works and how it could be used in many different fields as well. But if folks want to know more about your work, where would be a good place to find your research, your products and stuff that you're working on?
Sure. Well, they could search out my page at the University of Georgia, Wernell School of Forestry and Natural Resources, under my name, Dr. Caterina Villari, and my email is cvillari@uga.edu, so they can also reach out to me. I'll be happy to direct them to more information or a specific assays they need. But yeah, please reach out if you have any other question. And thank you very much for having me, Demian. That was very nice.
Yeah, absolutely. I'll talk to you soon.